You're good? Okay. So last week, during the pre-lab, you should have learned about the instrumentation and the software and how to calibrate the length and the force transducers and played with the stimulator. So now we're going to actually look at the length tension or force-length relationship of skeletal muscle to sort of quantitate its physiology. All right, we'll do this in one of the muscles of the frog leg. It's the semitendinosus muscle. I'll talk first about the frog preparation, what we're going to do for the dissection. I'll give you a demonstration as to how it's done, because it can be a little tricky to get this muscle isolated and then snipped out. And then I'll go through the 4 different parts of the laboratory exercise. All right? So the first step is going to be decapitation of the frog. And we're going to pith it, which essentially means once the frog's been decapitated, we take a probe and we kind of scramble up its brain stem and what's left of its upper spinal cord. When you're doing the dissection, the frog's arms, legs may move a little bit, and it's not to worry. The frog's not feeling any pain. These are just normal spinal reflexes, okay? It's not suffering at all, so don't worry about that, and don't jump, don't get afraid. The first part is to cut and peel away the skin on the frog leg. Just do one leg, leave the other one intact, because there's a very good chance, very high likelihood, if this muscle fails or the dissection gets butchered, then we'll go to the other leg, okay? We'll do a very blunt dissection to get to the muscle that we're interested in, okay? So essentially we're going to, somebody have a pair of scissors? Let me just, yeah, give me any. So when you're doing blunt dissections, you don't kind of snip, snip, snip at the muscle. You kind of go in with the scissors closed and then pull things apart, okay? There's a connective tissue casing underneath the skin that kind of acts as a sheath around all these quadricep muscles in the frog, and we're going to cut through that. And once we're through that, then we should be able to do a very blunt dissection with fingers to figure out where this muscle is. Once we have it isolated, we're going to put some sutures underneath it before we cut the muscle out, all right? And we're going to lay some omega hooks on it that are going to allow us to suspend the muscle in the chamber, and it'll also act as an electrical conductor. And we're going to suture those omega hooks right onto the muscle. Then we'll snip, snip, cut it out, okay? Once it's cut out, we'll measure its so-called resting length, L0, with a pair of calipers. Okay, just so when you get it into the chamber and you change the length, you'll have a relative idea how far away you are from what its resting length was when you cut it out of the leg. All right. The way it's going to be suspended in the chamber will look like this. All right. So here's the muscle, the omega hooks here in purple, and they've been sutured off with the little blue color suture. It's going to be suspended between 2 hooks. Right, one of these hooks is going to the displacement transducer that we played with last week, and the other hook goes to the force transducer. Now, connected to each of these hooks are going to be wires. They're soldered on, and sometimes they break off. If they do, let one of us know. We have a soldering iron ready to go to resolder these. These other wires are connected to the stimulator. Okay, this is the stimulator you also used last week. And this will allow us to, give pulses at different rates and frequencies to the muscle to stimulate it. Okay, we're going to fill this plexiglass chamber up with some lactated ringers. The manual, the handout says only 3/4 full. What's really crucial is not so much whether it's a half or 3/4 full, but the water or the solution, the level of the solution doesn't touch this omega hook. Okay, if it touches the omega hook, then when you stimulate the muscle, you'll get a substantial portion of the current not going through the muscle, but it can just be dissipated through the electrolyte solution. So however you move this, and you'll be able to move it up and down, you want to make certain that solution level is not touching the omega hook. Okay. It's plugged up by a cork that allows this bottom hook. It's got a hole in the cork, and the bottom hook can move up and down, and we put some Vaseline in there to sort of plug up any leaks, but it still leaks. You'll find out. So we have a couple of syringes of Vaseline that you can grab, and you can inject some Vaseline into that little hole to plug things up, because we want it to move with minimal friction, but we don't want it to leak. All right? When you have it suspended in the chamber, you want to pay attention to the length of the muscle, because it's going to be stretched a little bit more than the L knot. Whatever you use for the length, just be consistent. Probably the easiest thing to do is just to measure the distance between the two sutures. Okay? All right. Any questions on that? No questions on the prep. You're good. And I'll run through a prep so you can see exactly how we do this. Okay, so let's talk about the protocol for the lab. There's 4 parts to this, and we're going to look at 2 different types of contraction, isometric, and those are the first 3 parts, and then isotonic, or varying from isotonic to isometric in the last part. So an isometric contraction essentially means the muscle doesn't shorten, doesn't change its length. So it's trying to contract against more weight than it can lift. All right? To do this, once we've got the muscle in the bath, we're going to set our stimulator to a pulse of 1 millisecond duration and 1 volt intensity. And if everything worked out all right, we should see a little twitch. Okay? If you don't see it, just start increasing the voltage with this potentiometer, like we did last week, right? Okay? One thing you should pay attention to, the duration of the pulse is only a millisecond, but the A to D board will sample the waveforms every 5 milliseconds, 200 hertz. So there's a chance that when it samples the stimulator waveform, it may not sample right at the point where you see a pulse. Do you understand what I'm saying? So like I said last week, pay attention to that little lamp, the LED, on the stimulator, so you'll know that it's firing or not. And always remember to shut it off, especially if you have it on a continuous pulse train, because you don't want the muscle to get tired, get fatigued. Okay? If you see a twitch, keep increasing the intensity, the amplitude of the pulses, until you don't see any more change in the height of this mechanical twitch. And that's sort of where you want to operate. That'll be your operating voltage, plus or minus 20% or so. But you want to be right around the place where you get the maximum amount of mechanical tension. Okay? Once you have a nice twitch, just highlight it, copy it into an ASCII file like you did last week. All right? Any questions? Part 1? Straightforward? Okay, let's go to part 2. This is also going to be an isometric contraction, but now what we're going to look at is the effect of muscle length. Okay? So we started out at this L sub I, initial L, and what we're going to do on this displacement transducer, the little screw with a knob, we're going to turn it. And it doesn't have to be a lot, maybe 10, 15, 20 degrees or so. And we're going to vary the length of the muscle, or the distance between the two sutures, incrementally. So, and it'll be arbitrary. So we'll say L naught initially, L1, L2. And we're going to record a couple of different tensions. So one is a passive tension. Okay, and that's sort of its baseline mechanical tension. And that arises from the fact that intrinsically the skeletal muscle is elastic, just like an elastic band. And if you pull on it, it'll generate a force, just like a spring. Okay? There's another component in the muscle, is an active component, and when it gets stimulated, it'll generate force intrinsically, and that will be quantified by its total force. All right, this peak force right here. Okay, so you stimulate it, you'll record the peak force. This is defined as the total tension. Okay, and it's the sum of the passive tension and then the active tension. So to get the active tension, you can just subtract the passive tension from the total tension. Okay, and that will give you the active tension. Now, have people seen this diagram before in textbooks? This is theoretically what the length-tension relationship of striated muscle should look like. Okay, and it's divided into its 3 components: passive, active, and total. Okay, so you take a look. Let's look at the passive. As you increase the length, you're going to increase the passive tension. That makes sense, right? That's what a spring does. That's what an elastic band does. And that's what passive muscle will do. If you look at the active component, there's sort of an optimal length where the maximum amount of tension, active tension, the muscle can generate reach a peak, and we call this some optimal length L naught. All right, when you combine these two curves, you'll end up with a total tension that should look something like this. Okay, now whether or not your data looks like this will depend as to where you're actually starting in terms of your length. Okay, if your starting length is over here, then yeah, you could very well get a nice passive tension curve and an active tension curve that rises up and comes back down. But alternatively, you could be starting here. Okay, so your passive tension will keep increasing, but it'll look like your active tension is going down. Okay, doesn't mean you did anything wrong. All right, you just started at a different place. So if it doesn't look like this textbook example, that's fine. Just in your discussion in the lab report, you can explain why you think there's some deviation between your results and what happened. Okay. Questions on this? Nope. Okay. So let's look at part 3. And this is tetanic contraction. And what we do here is stimulate the muscle continuously, but we change the frequency of the stimulus. So ordinarily, at a single pulse or at very low frequencies of stimulation, the muscle will contract, develop some tension, and then it has enough time to passively relax down to its passive tension value. Okay? But if you keep increasing the frequency of the stimulation, you're going to reach a point where the muscle doesn't have enough time to go back to its resting tension. Okay? So what we're going to do here is, once we've played with part 2, we're going to return the muscle to its initial length, this L sub I. Set the stimulus frequency initially to 1 hertz and just record the resulting waveform. It should look something like this, hopefully. And then we're going to increase the frequency with that little slider bar. So we'll do 2 hertz, 4 hertz, 8 hertz, 10 hertz, 15, on and on and on. The frequencies that you're interested will be the frequencies where 2 phenomena occur. One is called partial tetanus, and the other one is fused tetanus. Okay? With partial tetanus, again, the frequency of stimulation is so quick that as the muscle starts to relax, doesn't have enough time to completely return to its resting value before another stimulus hits it. So what will actually happen is this tension curve will rise up and will sort of oscillate about some steady value, some mean value that'll be higher than its passive tension. Okay? And we call this partial tetanus. So you'll see some of these oscillations. Its mean tension will be a little bit higher than what it was at baseline. Okay? So you want to record that frequency. Then finally, fused tetanus, where the frequency is so quick the muscle has absolutely no time to even begin to relax. Okay? So it just has a constant level of tension. Okay? If you leave this on too long, what will happen is the tension will start to drop off with time. And you know why that occurs? You know what this is called? Anybody?
Rigor mortis.
Is what?
Rigor mortis.
Rigor mortis? No, I hope not. Rigor mortis, the muscle's not going to contract at all. And some of your muscles will certainly be in rigor. Is what? No. Fatigue. Yeah, this is fatigue. Okay? So what happens, the actin myosin cross bridges are cycling so rapidly, the muscle actually starts to move into anaerobic metabolism. The pH changes, intracellular pH, there's lactic acid produced, and the muscle just won't contract as effectively. And that's what happens when you get fatigue. So I tell you that because inevitably somebody's going to leave the stimulator on a frequency, a fused tetanus, and forget about it as you save your data, and the muscle's just going to be constantly stimulated. And that's a good way to really kill the muscle quickly. Okay? So you want to watch, always pay attention to what's going on with this little red LED. Okay? If you see that on, you want to make sure, if you don't intend to stimulate the muscle, make sure it's shut off. Okay? Where these frequencies occur is very arbitrary. I think Tuesday we were getting students that, I don't know, 14, 15, 16 hertz or so is where they were getting fused tetanus. Today it seemed to be a little bit lower, so it can vary anywhere from 8 to 20 hertz or so. So there's no really right answer where this is going to occur. Okay? Questions on this? So once you figure out what the frequency for your fuse tetanus is, you're going to use it for part 4. And this is probably the more complicated part of the lab, but I think this is the most interesting. This is where we look at the force-velocity relationship, all right? And the force that a muscle can generate, depending on how much weight it's pulling up or contracting against, how quickly it will shorten will depend on the load. So now we're not looking at isometric contractions per se as we did in the first 3 parts of the experiment, but this is going to be an isotonic contraction, okay? So initially, with the first 3 parts of the experiment, the muscle is going to be contracting on that cantilever beam against the baggie that's full of weight, so it can't shorten. It can only generate tension that's picked up by the force transducer. Now we're going to have just a little bit of weight in the bag, like 20 grams or so, just enough to offset its resting tension and the weight. And we're going to add a little bit more weight each time we do this. Okay, so say start off at 20, 30 grams. And the frequency that you found fused tetanus, whatever it is, 15, 20 hertz, you're going to use that frequency to stimulate the muscle with the 20 or 30 grams in the bag. And you're going to record the length and the force strips, okay? Don't leave the tetanic stimulation on too long. Just hit the button once, get these tracings, and then shut it off, okay? What you're interested in for a given load is the velocity of shortening. And you can get that by examining the length tracing. And you can zoom in terms of the time scale as to, this piece right here where the length is changing is a function of time. You want to highlight that. And there's an option in the BioPack software so you can compute a derivative or a slope of that portion of the waveform. So you will get the derivative or the slope of this waveform, delta L over delta T, and that's an estimate of the shortening velocity. If you've calibrated it, it should be in millimeters per second, okay? So you compute the velocity, and you just need to write it down in a notebook. 30 grams, my shortening velocity is this, and at 50 grams it's this, and at 70 grams it's this, and you go back and forth. Now what happens to velocity as you increase the load that the muscle has to contract against? Goes down, right? How does it go down? Does it go down linearly? Yeah, you could describe it exponentially. The model that they use is hyperbolic, okay, which implies that the product of force and velocity is constant, okay? And when you see that, the assumption is that the muscle is generating constant power, okay? Whenever you have a through and a cross variable, whether it's force, velocity, current or voltage, pressure or flow, if the product of those two are constant, it means the power dissipated is constant. Okay. Ideally, it would look a nice, beautiful curve like this. When you have zero shortening velocity, right? What kind of contraction is that? Isometric, right? And then as you decrease the force or the load that the muscle has to decrease, you're going to start to increase the shortening velocity. Okay, it's not going to look perfectly hyperbolic, but there are functions in Excel, I think, where you can fit a hyperbolic curve to the data set. I'm sure there'll be some scatter, but you want to see. And if it doesn't look like this at all, just come up with an explanation as to why you don't think it doesn't. Okay, but this is the model. This is the assumption. Is constant power. Okay. Any questions? No? Okay, so here's what we're going to do, because I have to be filmed for this. I guess Marty's going to cut Frog's head off, give it to me. I think this group's small enough that most of you can gather around. I'll do a couple of these as well so you can see how I do the dissection. But we're going to do one to put on the tape. Okay? Questions? You're good? All right. Let's go cut.
Go.
Okay. So the first part is to cut and peel away the skin of the frog's thigh. That's what I'm doing here. Okay. All right. And you can see underneath the skin there's this connective tissue sheath. That's just in fascia. I'm going to do a blunt dissection. Okay. So I'm really just pulling apart those connective tissue fibers so I can kind of get down deep with my fingers to find this muscle. The muscle that I'm looking for is the semitendinosus muscle. It's got two heads, sort of in the posterior, the rear part of the thigh. Okay, I think this will probably be good. Oops. It's not popping out at me. Okay. Okay. And here it is. Okay. See the two heads right there? Okay. One, two. They kind of fuse together. And what I'm going to do is take a piece of suture and I'm going to put the suture underneath this muscle. So grab it with a pair of pickups. Okay. And it's important to do a square knot to tie these omega hooks on. And the omega hooks, you want one loop pointing proximally this way and the other loop is going to point distally. So before I throw that knot down completely, I have the little omega hook and then I go ahead and I tighten it the first throw. It's going to be tight but not so tight that I'm going to cut the muscle fibers. And then I put a throw in the opposite direction. Okay? And I would do 3 or 4 throws. All right, and that just minimizes the likelihood that the hooks are going to come off when you have this suspended in the bath. Okay, there's one. And this is going to be number 2. So I get my other omega hook, put it in there, tighten, but not too tight. 1 throw, 2 throws. Okay. All right, so they're both on. Now I'm going to go ahead and cut this muscle out of the leg. All right. And there you have it. Now the other thing I do, just to make certain that these hooks are tied on tight enough to the muscle, is I grab them between 2 sets of pickups and I just put it under a little tension to see if that'll hold. Okay? And that looks pretty good. I think that's going to do fine. Okay? For now. It may still fall apart, but right now I think this looks pretty good. Okay? Then you can go ahead, you can measure the distance between the 2 sutures with a pair of calipers. Whatever it is, you got one of those? Yeah. Okay. So, go ahead. Probably this way is fine. You can go ahead and measure that. Then when you get this suspended in the bath, you can do the same thing. So you can just get an idea of the difference in the amount that you stretch the muscle. Okay? Any questions? I'm going to do another one, so this was just for the videotape.
Do we need to measure diameter?
You don't need to. I know it says you do, but the diameter really depends on how much the muscle has been stretched, and it's pretty variable. So I would just record the forces, and you don't need to normalize it by the diameter. Okay. All right. I think that's it. Questions? Okay. Who wants this one?